In this video, we're going to talk about enzymes and potential energy. And the first key concept in all of this is that chemical bonds themselves contain potential energy. And when you break those bonds, you can release energy. And conversely, when you form the bonds, you're going to have to put energy into the system. Right? And so these terms, anabolic and catabolic, get used a lot in biology and biochemistry. And anabolic really just means that smaller starting molecules are going to be combined to make bigger ones. So you can think of a lot of different examples for this. These could be molecules of glucose, for example, and the polymer that we build from them could be something like starch or glycogen if we're in an animal. So the concept is you're going to have to add energy to make that happen. Glucose molecules don't just randomly start forming starch or glycogen. You'll spend energy to make that happen. In a biological setting, that energy usually comes in the form of ATP, which we'll talk about in a bit. All right? Later on, when you take those complex molecules and you break them apart into smaller ones, that is catabolic. You're just doing the exact same thing in reverse. So that would be taking our starch or glycogen and breaking it back into glucose. Right? And this concept works for any biomolecule. So this could just as easily be amino acids being converted into a protein. It could be fatty acids being converted into triglycerides, nucleotides turning into DNA and RNA. Anytime you're taking small starting molecules and building them into something larger, it's anabolic and catabolic. Right? For the sake of things that are going to come later in the class, anabolic is a term that's usually used in biology. In a chemistry term, this almost always means endergonic. And endergonic just means requires energy, whereas these catabolic reactions are almost always exergonic, which just means they release energy. Right? And so we'll pair those terms together in a bit to try to make it make more sense. Right. Another concept that we need to have some understanding of is entropy. And entropy really just means disorder or randomness. And that just means that as the more molecules spread out, as they break down into more pieces, as things become disorganized, entropy goes up. And the concept is the universe wants entropy, right? Everything becomes more disordered over time because randomness starts to take over. And so one way of thinking about it is life in general really just means avoiding entropy, avoiding disorder. Right? A human or a tree or a fish or any organism, even bacteria, they are very ordered structures. We have cell membranes, we have proteins that are assembling together. And so life really just means fighting entropy. Right? In their example, if you take a substance that's a solid and you convert it to a liquid, entropy goes up because in the liquid, the molecules get to move around more, they're more random. If we converted that liquid to a gas, and the molecules got to spread out even more, that would be even more entropy, right? Going back to these examples, if you are going through anabolic reactions, you are decreasing entropy, right? Because the larger molecule is more ordered, it's structured, right? When you're broken into a bunch of little pieces, that's the disordered version. And so the reason that anabolic reactions require energy is that we're fighting entropy. And the reason that catabolic reactions tend to release energy is because they're going the direction that entropy wants them to go. We're increasing the amount of entropy in the system. Okay. So putting some of those concepts together, if we think about how cells use energy, this molecule ATP is the fundamental unit of energy currency in a cell. And all that means is when you take in high energy molecules like glucose or fats, we'll use glucose as an example. One glucose has the energy of dozens and dozens of ATP. It's an inefficient process, but most organisms in most circumstances get about 32 ATP out of glucose. Plus, they also release some heat because you don't get 100% energy transfer. Okay? And so when we say that we're converting the energy from glucose into ATP, all that means is we have this adenosine molecule, right? This is the exact same adenosine that's the A in DNA, like A, C, T, and G, base pairs. So you've got your adenosine base, you've got a ribose sugar, and then we've got 3 phosphates. So this is ATP for adenosine triphosphate, tri like 3. If you removed one of these phosphates, you would have ADP, right, diphosphate. And if you remove the second phosphate and we're down to 1, this would be AMP, adenosine monophosphate, mono for 1, right? So in all of this, the chemical energy is being stored in these bonds between the phosphates, right? And so when we say that we take an ATP molecule and we use it or spend it, that really just means that you're taking this ATP, breaking the bond between the second and third phosphate, and this phosphate floats off by itself. It's usually written as PI for inorganic phosphate because it's not attached to anything with carbon anymore. And then the ADP that you have left behind has a relatively low energy because breaking that bond released that energy that we want to do some work, right? So breaking that bond in ATP, that is an exergonic reaction, and it's catabolic. Right? We're breaking down the ATP, releasing energy, and that energy can be used to do a lot of different things. It could be used for a muscle contraction or to run an active transport pump, like we're going to see in a little bit. But we're harnessing that energy, and the reason we converted glucose's energy into ATP is just so it's in a usable size that's not too big, right? Using an entire glucose at one time is like lighting a stick of dynamite when all you really need is a firecracker. So we're breaking it into smaller pieces. Okay, so an example of how that energy from ATP could be used, right? This sodium-potassium pump, that is an example of active transport, right? Which just means that we're moving ions or molecules across the membrane, and we're moving them in directions that they wouldn't naturally want to go. So in this case, we are pumping sodium outside of this cell, so this would be the extracellular space out here, and we're pumping potassium inside of that cell. The catch is, remember, normally ions like to diffuse from high to low concentration, right? So if this was pure diffusion, we would expect everything to always diffuse only from high to low concentration. Here, we get to use ATP like a pump, so we can drive these things from low to high. That gets us a very high concentration of sodium outside the cell and a high concentration of potassium inside the cell. Why this matters will come into play later on in the class when you eventually hear about neurons and how they function. But these gradients, sodium on this side, potassium on that side, is a huge part of how neurons in the brain send electrical signals. Right? Because eventually when we open a diffusion channel, these ions are going to flow down their gradient, and that's going to be a signal that gets sent. So none of this would happen if you didn't put ATP in the system, right? Because we're fighting entropy the entire way. Right, so to tie this into a few of the terms that I've been using, exergonic and endergonic, you will see graphs that look like this in bio classes and definitely when you get to a general chemistry class. This concept of free energy is just asking the question, how much energy, potential energy, is in our molecules, right? And so in this graph, the y-axis is free energy, how much potential energy we have, and the x-axis, we're showing the progress of the reaction happening over time. So remember, in chemistry, the reactants just mean whatever molecules you started with, and the products mean whatever molecules you ended with. And so all we're saying in this case is the reactant started off up here, and the products ended way down here. And so the difference between those two, we call that our delta G. It stands for Gibbs free energy. And the delta sign, that triangle, always means change in. So delta G just means change in free energy. And in this case, our free energy decreased by that much. Okay? Since delta G went down, we say that it has a negative value. And that's the definition of an exergonic reaction. A negative delta G means that you released energy. It usually also means that you increased entropy. And these are reactions that happen spontaneously because molecules naturally want to go to their lowest energy state. All right. The second concept in all of this is you have something called an activation energy. Right? So our reactant started here, and imagine that over the course of this reaction, it's kind of like pushing a ball up a hill. Right? So this is our ball. We're trying to push it over the hill, and if we could just get it up here, right, it would very spontaneously roll down the hill because gravity would take it down. The activation energy just means that you have to put some energy to get it to the top of the hill. In chemistry, it really means that you're putting energy in to start breaking those chemical bonds. They need a little bit of a push. So a good example of this is anytime you're lighting something, like imagine lighting a candle. The candle doesn't just erupt into flames spontaneously. That wouldn't happen. You have to give it a push, an activation energy. And so lighting your match or your lighter and holding that flame up to the wick, that is pushing the wax molecules in the candle up to their activation energy state, and the reaction starts going. Now, as soon as the reaction starts going, the candle starts making heat. And so the heat that's given off, right, delta G, we're releasing energy, that heat can then come around and fuel even more reactants, more wax, reaching its activation energy and proceeding. Right? Now, notice the activation energy does not have anything to do with delta G. For example, if we add a catalyst to the mix, right, when they say a catalyzed reaction, a catalyst is just something that makes a reaction go faster. It can't make reactions happen that wouldn't otherwise happen, but it speeds them up. And so if you add a catalyst, your activation energy goes down, which makes it easier to get our ball over the hill, so the reaction speeds up. But the delta G doesn't change because our reactants and our products are still exactly the same. That value is still the exact same number from the first reaction. All right? This will matter in the next video we go over because enzymes act as catalysts, and they're what control the vast majority of biological reactions. So watch out for that in the next video. Okay, on this side, if we have an endergonic reaction, it's the exact same concept, but now delta G is going to be a positive number. And that just means that our reactants started off relatively low, and the free energy of our products, right here, ended up relatively high. Right? And so you still have that activation energy, right? Push your ball all the way up the hill, and then it rolls down a little bit, but it still ends up higher than it started. Right? And so over here, products were lower than reactants, negative delta G, spontaneous. These things generally want to happen naturally. The enzyme is just going to speed it up. On the endergonic side, these things do not want to happen naturally. Right? Your products end higher than the reactant started. These are reactions that are typically decreasing entropy. Right? And to link this back to the concepts that we talked about in the beginning of this video, these exergonic reactions tend to be catabolic. We're breaking things down. So think breaking apart sugars and amino acids and fats to get energy out of them, right? Like glycolysis and citric acid or Krebs cycle. And endergonic reactions, think any sort of synthesis, right? Protein synthesis, glycogen synthesis, making fats. Anytime you're building something complex, it's going to have a required energy input. Okay? And to link it back to ATP, think about these catabolic reactions as like breaking down things with calories, like our glucose that we've been using as an example, and you use that glucose to make ATP. And then what does that ATP do? You use it to fuel those synthesis reactions. So all we're really doing is extracting energy from exergonic reactions, right? Using glucose, fats, proteins for an energy source, converting the energy into ATP as an intermediate, and then using that energy from ATP to fuel endergonic reactions that wouldn't happen spontaneously. And this is how we decrease entropy, right? Take energy from our food and use it to drive these anabolic, right? These would be synthesis or anabolic reactions that need energy in order to happen, right? And so this is really just energy conversion. Every organism that eats their food has to do this, right? Take in food with energy, extract it over here, use that energy to make reactions happen that wouldn't naturally happen, right? Because they're endergonic. Okay. All right, I hope this was helpful. In the next video, we're going to go into the details of how enzymes alter activation energy, how they speed up reactions, and how we regulate them. So make sure you keep track of the order of the videos in the playlist, and please subscribe. It'll help you keep track of all the videos that I post, and eventually we'll get through a lot of topics that cover everything, hopefully in the class that you're in.